Live long and prosper.
Cells are the smallest unit of life. We are made up of trillions of cells, and inside each one is our DNA. If you were to stretch out all the DNA of one cell, it would be over 2 meters long. In order for your DNA to be packaged into a tiny cell, the strands are wound around special proteins called histones. These help consolidate the DNA by supercoiling, kind of like if you were to wind a telephone cord around itself. Once it is completely coiled, the DNA forms chromosomes. These are the structures that you may be more familiar with. The ends of chromosomes are called telomeres, which are composed of 6 bases, T, T, A, G, 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 repeated thousands of times. You can think of a telomere as a little piece of plastic at the end of your shoelace, which is called an aglet. Without an aglet, there will come a point where your shoelaces are too short to tie together. Thankfully, this plastic cap prevents your shoelaces from deteriorating, just like a telomere is present in order to protect the ends of your chromosomes. When the cell is ready to divide, it needs to replicate all of its DNA. Unfortunately, the process of replication means that a small part of the telomere is lost each time, about 50 to 200 bases per cell division. If you lose too many bases, you run the risk of losing essential genes your body needs in order to thrive. If we have this potential problem of losing genes, and we start from a single cell, how did we get to be made up of trillions of functional cells? It turns out that some of our cells can actually repair their telomeres. This occurs in our stem cells and in our germ cells. Let's take a look at that. Here we see your typical telomere after replication. Notice the overhang on the 3' end. Now notice that there are free-floating bases constantly surrounding the DNA. Telomerase sees the overhang and, using its own RNA as a template, will take free-floating bases and extend the leading strand. Primase then puts down a complementary RNA primer. This allows DNA polymerase to restore the missing nucleotides. Finally, polymerase leaves, and so does the primer. So what about the rest of your cells? The cells that make up a majority of our body, somatic cells, stop dividing after about 50 times, and the oldest cells begin to die off. This is what causes us to see physical changes as we grow older. Wrinkly skin, worsening vision and hearing, and gray hair are all results of cell death. So why don't all of our cells use telomerase? We could be immortal. While this sounds great, it is more likely to result in cancer, which is when cells over-replicate and avoid being destroyed. Over 90% of malignant cancers have overactive telomerase, which is how they are able to grow so fast. Luckily, scientists have begun to target telomerase in new cancer therapies. One of the more promising solutions is injecting an inhibitor into the cancerous mass, which stops the overactive telomerase activity. This halts cancer growth, increasing chances of patient survival.